This is a special webinar presentation tonight. We're being funded by the Massachusetts Department of Agricultural Resources, otherwise known as MDAR. They are the funders for this webinar and for the grant that you'll hear more about tonight and on December 1st, when you will listen to part 2 of this series. This is part 1, dealing with mycoremediation, bioremediation, an introduction to that. And then on December 1st will be our part 2 actual online event that my colleague, Doug Cook, would be hosting, where you get to see our work firsthand here in the city of Springfield. So please tune in for that. You don't want to miss that. Another thank you I'd like to give out is our partners for this grant, Gardening the Community, as well as Home City Housing. They are the on-the-ground partners here in Springfield that are working with NOFA on this grant. And a big thank you to our consultants that are assisting us. Nancy Clem of Spontaneous Vegetation and Social Ecology, Nathan Harmon of Advancing Equal Agriculture, Willie Crosby of Fungi Ally, our presenter for tonight, and Leah Zelbalik, an environmental scientist and bioremediation specialist. We are very grateful to the consultants who are helping us during this 3-year grant. Now, as I mentioned before, the webinar is part of a 3-year grant project funded by MDAR, and its goal is for community-based urban agriculture organizations to learn about and trial natural bioremediation methods. So with no more from myself, I turn everything over to Willie. Willie, thank you so much for being with us tonight.
Yeah, thank you, Anna. It's great to be here, and I'm excited to, as always, excited to talk about fungi and mushrooms and their role, in the world, and I guess particularly this time in the world of mycoremediation and helping to create an environment and a world that we all can live in and can help be healthy in. I always like starting with a little breath to kind of bring us together. So an invitation, if you're comfortable, to just close your eyes and feel your feet on the ground, and just bring your awareness to your nose, to the sensation of the breath passing in and out of your nose. And notice the quality of your breath. Is it shallow or deep? And maybe just relax into this moment, into being in this place on the earth, standing, sitting, in this body, with all these other people. Just relaxing into being here, allowing yourself to be present here. So thank you. When you're ready, you can open your eyes. Great. So, okay, I'm going to share my screen, and I guess a little bit about me, where I come from, why maybe I'm talking tonight. I started growing mushrooms back in 2013, and they totally hooked me. You know, I was actually working with Caro at Simple Gifts Farm in Amherst, Mass, doing a CSA vegetable operation, and the mushrooms just like got me and pulled me in, and I started growing mushrooms at my house and read an awesome book, Mycelium Running, and was really inspired by not only mushrooms as a food source, but all the different ways that they can benefit us as medicine, as mycoremediation partners, all sorts of different opportunities to ally with fungi. And what I really focused on was cultivation. So I started growing more and more mushrooms, and in 4 years or so, I had moved out of the basement of my house into a warehouse, and was growing about 500 pounds of mushrooms a week. shiitakes and oysters, lion's mane and piopino, lots of different mushrooms. And during all that time, did a lot of education work and a lot of exploration, because there's so much to explore with what mushrooms have to offer and how we can work with them. So it's kind of this constant. Mycology is an exciting field because there is such a exploration of it, of what can these organisms do and how can we work with them to improve ourselves and the world around us. Um, so, I started to move in the last 2 years. I've started to move more in that direction, empowering other people to grow mushrooms, selling grow kits and spawn, and doing more research into the medicinal aspects of mushrooms, how to access those, um, and the power of mycelium in general, and what does mycelium have to offer beyond just a food source. Um, so and I like love mushrooms. I don't know if people have seen the cordyceps video on BBC, where the cordyceps like infects the brain of the ants, and for me it almost feels like mushrooms somehow have infected my brain and have like taken over what I do, and are just like, do mushroom things, grow more mushrooms. Um, so they're in me, and I work with them every day, whether that's through a tincture or food or cultivation. But they're very much a part of my life. Um, so that's some about me, and now we can really focus in on the fungi, and I can share with you some about mycelium and what they are. It's always important to kind of start at the base level of what fungi are, and what they do, and the general life cycle that mushrooms follow, because oddly a lot of that stuff is skipped in the, I don't know, school realm, in the education realm. um, First off, what is mycoremediation? The word is a combination of two words, myco meaning fungi. So myco, you might see myco in front of a lot of different words, and it's just being like, oh, this is a mushroom related thing. Um, so mycorrhizal is another really common place you see that, and that's myco fungi rhizal root. Um, and then you'll see it in more comedic places, like I don't know, nothing's popping in my head, but um, yeah, you can just say it for anything. Myco movies, myco packaging, myco building materials, whatever. And then remediation basically is the reversal of environmental damage, and a lot of times that's what we're focusing on is a human implemented damage. So mycroremediation, the use of fungi to try and reverse some of the environmental damage that we have caused on this beautiful earth. So it's important to first understand what are fungi. And fungi are their own kingdom, separate than plants, separate than animals. And there's three things that classify an organism as a fungus. Their cell walls are made of chitin, their mode of digestion is absorption, and they're heterotrophic. So heterotrophic meaning they can't produce their own food. They don't photosynthesize. So just like us, they rely on an external food source in order to survive. They're not like plants where they can make food out of the sun. So their food source can be plant, like they can be saprophytes, so they can eat dead material. They can be parasites, so they can eat living things. And this can be plants, it can be animals, it could be bacteria, it could be nematodes, pretty much anything that's alive and dies. Fungi are involved with consuming. And that that also points to fungi's relationship with light. And it's important to know that since they're not photosynthesizing, light isn't crucial for fungi to thrive and live. Um, Mode of digestion is absorption. What that basically means is that they have an external stomach. So as they're growing, particularly the mycelium, and we'll talk about what mycelium is, but the mycelium secretes enzymes outside of its body, breaks down material, and then sucks nutrients back into its body. So it's actually able to leave some of the waste products outside of its body. And it also has to grow in its food source, right? So because we have an internal digestion system, we can walk around and grab food and eat it and then walk around more. But because they have to be like touching their food at all times, they actually grow, in their food. They become their food. And it's those chemicals, those compounds that they're secreting that are primarily what's used in mycoremediation. So whatever those compounds they're excreting to digest their food source is what can break down these pollutants. And lastly, chitin. This is interesting for eating mushrooms. It's chitin we can't really digest. So about 95% of people can't break down chitin. So if you eat raw mushrooms, you can't get any of the medicinal or nutritional benefits that they have to offer. You have to break down that chitin before they're available. Some more info about the fungal kingdom. There's a huge range of estimates of how many species there are, anywhere from 1.5 to 8 million species. And a rather small percentage of those, 10% or less, actually produce mushrooms. A mushroom would be like a fruiting body, like you can see down here in the left-hand corner. A fruiting body that can be picked by hand and seen with the naked eye. So most fungi don't produce mushrooms. And it's, where we're most familiar with fungi. I guess there's some non-mushroom producing fungi that we're familiar with, particularly plant pathogens. But where a lot of mushrooms are researched, or a lot of fungi are researched, are ones that produce mushrooms. And a lot of the ones that are cultivated are ones that produce mushrooms. So there's this whole realm of fungi that don't produce mushrooms that we're really just starting to understand what they do and the benefits that they have to offer. And we know a really small amount of those fungi that produce mushrooms. It's up to about 18,000 at this point. But again, like 12, 15% have been identified. So a really, really small percentage. And the conception of, or like the fear of mushrooms isn't based on the numbers. It's not like there's way more poisonous mushrooms than there are plants. Actually, the amount of mushroom species that would kill you are pretty low. So I think it's more just difficult to become familiar with mushrooms and in identification, because an oak tree is there all year round and has all these, you can use the leaves or the bark or all these different ID features and you can get familiar with it. Whereas a mushroom comes and is gone, it can be in as little as 24 hours or maybe 2-3 weeks. So it's a lot harder to get familiar with mushroom identification. So fungi is the kingdom, and then below that we have phylum, and there's 13 different phyla in the fungal kingdom. And I want to talk about the life cycle of one. Two of those phyla, the Ascomycota and the Basidiomycota, produce mushrooms. The other 11 don't produce mushrooms. So I want to talk about the life cycle of the basidiomycetes, the ones that produce mushrooms. And essentially there's three parts to it. You have the mushroom, which one of my teachers likes to call a temple of copulation. It's a place that sexual reproduction happens. And the whole point of it is to produce spores, right? And spores go out, they germinate into mycelium. So those are the three key things to know: mushroom, spore, mycelium. There's a lot of different ways to identify mushrooms, but one of the first kind of like key characteristics is, what are the spores produced on? So this is a pink oyster mushroom, and a bunch of gills here, and that's where the spores come out of. And that could be gills, tubes, pores, spines. It could be just a club. Um, uh, Corals. There's a lot of different ways it can come out. But the spores are on these mushrooms by the thousands and thousands of spores on that. And spores are microscopic unless you have them in mass like this. So this was a pink oyster grow kit. You can see exactly where the grow kit was. And there's tons of mushrooms on the side of it. And overnight, those let out all these spores, right? So there's tens of thousands of spores on this desk, and you can see it at that point. And the reason they make so many of the spores is because they are microscopic. They're going out, they're being blown in the wind and disseminated by beetles and rain, and hopefully they're landing in a place that they can grow again. It's like, this is the way that the fungus travels, is through the spore. So they're just going out and playing the numbers game of like, I hope I land on a conducive substrate material that they can grow on, and I hope I land close to a compatible mate, someone that I can sexually reproduce with. So they produce tens of thousands of spores. There's a mushroom called the artist conch, and a mature artist conch can be like, twice the width of me. And in a single year, if you took the spores and laid them end to end, these microscopic spores, they would reach around the world. So it's incredible, the quantity of spores that are being produced. And then we get to the real magic. The spores germinate into mycelium. Mycelium has one cell wall. It's very susceptible to like environmental changes and damage. It's really susceptible to drying out. So you never see mycelium. It's like this all pervasive, super powerful thing that's all around us, but we never see. And if you like flip over buried logs or rotten logs, or dig into the leaf litter some, then you'll see this like white cottony mycelium. And that's how fungi spend most of their lives. So all the 11 phyla that I was talking about that don't follow this exact same life cycle, they all have mycelium, and a lot of them you'll see as mycelium. So like the bread mold that you see, if you just leave wet bread on the counter, or even if you have something in the fridge get moldy, that's mycelium. That's mycelium that's growing up and kind of making it look that like fluffy cotton webby thing. So a lot of what it does is break down dead material. It's the mycelium is the reason that we're not just totally buried in dead organic matter. It's continuing to promote the like process of things dying, to basic nutrients, to things living. So it facilitates that death to basic nutrient blocks for the next thing to use it as life. And there's tons of different compounds that the mycelium is secreting out into its environment. A lot of them are digestive compounds, but they're also antibacterial compounds, other antifungal compounds, all sorts of different things. And for mycoremediation, the fungi that are really being studied are white rot fungi. And they're fungi that break down both lignin and cellulose. So cellulose is a pretty basic compound for things to break down. It's very close to a sugar. So a lot of things can break cellulose down, but lignin is a complex hydrocarbon. It's really difficult to break apart the bonds and to make it, accessible to break down. And the white rot fungi can break that lignin down. And it's interesting because lignin, a complex hydrocarbon, is very similar to petroleum products. What petroleum products are complex hydrocarbons that nothing knows how to break down. And fungi can have the tools, have the enzymes to break that stuff down, which is totally, totally fascinating. So here we've got a great example of a wine cap mushroom. This is a mushroom. It grows really, it's native in the Northeast. It's pretty prolific. You'll see it in wood chip piles, like playgrounds or backyards, or sometimes leaf litter. It has this partial veil on the stem, grows in clusters or singularly, and then it has this really tenacious mycelium. So a couple things that wine cap have been used for is erosion control, because it can just grip onto stuff so ferociously and just doesn't let it go anywhere. And also cleaning of wastewater. So that mycelium is really good at filtering bacteria and other fungi. So you have bacteria like E. coli or staph, or even like sediment, like if you have stormwater runoff, having a patch of mycelium, of wine cap mycelium, really helps to catch that stuff and break it down. And it eats it. It doesn't just like kind of catch the E. coli and the E. coli grows there. It actually catches the E. coli and then consumes it. So working with this mushroom, and in the backyard, you can do that. If you have a composting toilet, or if there's like runoff in a certain place that you're concerned of a bacterial load. Yeah, they're totally delicious as well, wine caps. Yeah, a question about what about heavy metals, and we'll hit on that in a little bit, but mushrooms are not good for heavy metal take-up in general. So here you have an example of an oyster mushroom. This is microscopic. It's an oyster mushroom mycelium predating a nematode. So oyster mushrooms actually make these little like lassos out of their mycelium. You can see two right here. And a nematode swimming along in the soil, and it hits one of these lassos, and then the mycelium grips it, right? And then it slowly grows up into the mycelium grows into the worm and just completely eats the worm. So an incredible, it's like this whole microscopic world that's going on, and mycelium is a big player in consuming things like nematodes and bacteria. This is an example of Paul Stamets' work with oil. So what he did was worked on 3 piles of soil infected with oil. And there's a control pile, there's one with plants and one with oyster mushrooms. So the spent substrate, essentially the spent substrate is after like a mushroom farmer grows mushrooms, there's sawdust and mushroom mycelium that they're just going to compost. And you can take that mushroom mycelium and use it to inoculate another substrate, to plant the oyster mushroom into another material. So a bunch of spent substrate, oyster mycelium on sawdust, was added to this pile of oil infected soil. And after about 4 weeks, the mycelium had grown into the soil and was feeding off of the oil, breaking down the oil, and making these massive flushes of oyster mushrooms. I mean, this whole thing is just fruiting huge flushes of these are like bigger than your head, these oyster mushrooms, So it's thriving on this oil-based soil, which is absolutely incredible. And they did tests looking at the mushrooms, and there's no oil in the mushrooms. It's actually breaking down the oil into basic building blocks and then making nutrients for it to grow. And it decreased the oil to incredibly healthy levels. And this is the same pile about 2 or 3 weeks later, and there is a pretty healthy insect and worm population starting to grow, and all these plants starting to grow. And the other piles just looked like, dead, totally dead soil. There's nothing happening on them. These brown things are the dried up mushrooms that you see here. So just in like 6 weeks or so, the oyster mushroom mycelium was able to convert this totally dead, nothing can grow on a pile of soil into a space where there's insects, there's worms, there's plants, there's like, wow, this is now becoming on its way to being a healthy ecosystem. So mushrooms and fungi are an awesome ally in the breaking down of oil, diesel fuel, and petroleum products in general. On a small scale, oil is all around us. If you're someone that changes their own oil with a car, or if you have a bike and you get bike grease or bike oil, or even if you have a bike shop in your neighborhood and they're wiping bikes down with oil, those are all things that you can just be like put in a bucket or something and add oyster mushroom mycelium, and it'll just consume that oil and turn it into healthy soil. So this is an example, just kind of seeing like an experiment looking at what the oyster mushroom mycelium does. You have straw here with the oyster mushroom mycelium grown on it, and pouring oil into that straw. And it just grows up over 2 weeks and just eats the oil as a food source. So an incredible capacity for remediation of oil and those sorts of products. 3 of the most studied mushrooms that are used in mycoremediation are oyster, wine cap, and turkey tail. All are really good at breaking down either. Oyster is such a ferocious mushroom. It grows very fast. The mycelium grows very fast, and it can consume a wide range of material. So that's a really good one when you're looking at mycoremediation. And wine cap has been looked at mainly for more like living organism contamination, so that bacterial or fungal or nematode sort of contaminations. And then turkey tail has been tested a lot with pesticides and nerve gas and those sorts of maybe less well, at least with nerve gas, very much less like prevalent. But of course, pesticides are all over. And then there's tons of non-mushroom producing fungi that we've looked at in terms of being beneficial for mycoremediation. So this is a list of some of the different products that mushrooms have been used for mycoremediation. Atrazine is a really prevalent pesticide, and actually Trad Cotter's done a lot of work with growing mushrooms on atrazine and has been able to grow mushrooms on a petri plate of just 100% atrazine. All the mushrooms we're eating was this super toxic pesticide. And you can train mycelium. So what you do is you can train mycelium to eat this sort of food source. So if you have a Petri plate and you add 20% atrazine and then have 80% sugars, it'll eat those sugars and then slowly start to eat the atrazine. But the way it works is, fungi kind of have like this keychain of compounds that they can produce. And when they run into a new food source, they're like, okay, what key breaks this down? So they're starting to try all these different keys to be like, does this compound break that down? Does this compound break it down? And then once they find out what compound breaks down their food source, whatever that is, they can say, okay, send a message through the mycelium. Okay, here's the key that breaks this down. Let's start making that. And then they just take off and start eating it. So you start out at 20% atrazine and they figure out, oh, here's what we need to produce to break this down. And then you can have the next generation be 50% atrazine, and then the next generation 75, until you're finally at all atrazine, this crazy pesticide, and they're having no problem breaking it down and consuming it. So there's this ability to train mycelium and work with mycelium to break down specific um, pollutants. Oil, as I was talking about, really good. And then other petroleum products as well. So plastics and other petroleum-based materials. And then to kind of hit on the heavy metals piece, what mushrooms do with what fungi do with heavy metal is they hyperaccumulate them into the mushroom fruiting bodies. So they can't, heavy metals can't be broken down, so they're just moved around. And so the mycelium moves the heavy metals out of the soil, takes it up, and then puts it into the fruiting body. And plants do this as well. And the difficulty with mushrooms is that they fruit and then they're gone in 3 days or a week. So it would take a lot of monitoring to get the mushrooms and be able to remove the heavy metals. And of course, the mushroom is then a contaminated source. Whereas with plants, you can have a plant growing and it's there for 6 months, and you can just be like, okay, I'm going to go harvest the plants, in 2 or 3 weeks, and remove the heavy metals in that way. So for heavy metal contamination, plants are much better situated to remediate that because they are there all the time. With mushrooms, it's kind of like they'll probably hyperaccumulate it into the mushroom fruits, and then it dissolves, and then it just goes back into the soil, and the mycelium picks it up again. It kind of becomes this cycle, but there's not really anywhere for the heavy metals to go. So it's a very different thing than the oil or the pesticides, or the bacteria, where the mycelium is actually eating those things, breaking those things down, and turning them into something that other organisms can use and live off of. So those are some of the ways that mushrooms and fungi are being used in mycoremediation, and some of the potential that we have to work with them. I think one of the big places that there's opportunity is, okay, how do we know these things are possible? How do we get them into the implementation stage? And a big big benefit with working with mushrooms in this way is that it can be done on site. So a lot of remediation practices remove soil off the site and break a contaminant down, and then bring new soil in. And with mushrooms, we can be breaking down the pollutant right on the site and don't have to be removing soil. And in fact, we're adding soil, we're adding organic matter. So it's like, okay, let's make the soil that we have healthy, rather than removing it and bringing it somewhere else to break down. Um,
Willie, can I interject a question?
yeah, please.
So a couple of questions came in. I think you may have addressed it as it relates to heavy metals, but there's a curiosity about what it could do with lead and plastic.
Hmm. Yeah, the lead and arsenic and cadmium, all those sorts of things. Mushrooms just aren't the ideal thing for
Hmm.
it. It's really the plants that I think are the best allies for those heavy metals. Really, where mushrooms shine is with the, yeah, plastics, oil, herbicides, pesticides, all those sorts of things where the mycelium can really break it down into basic building blocks and get it out of the soil, get it out and start bringing the plants and the insects and the worms back into the soil, making it a habitable space for other organisms. So yeah, there was a team, and so much of this is on the lab scale, so there's some, but there's pretty few implemented microremediation projects, and that's part of what I'm excited about this project. And I think, as more and more people learn about microremediation, it'll be hopefully implemented more. um, but to really see what can they do in the field, and how can we, apply this in a city lot, or in our backyard, or in our neighborhoods. I think finding those systems is really important. um, in terms of, can they eat plastics, yeah, there's oh, what is it, I can't remember the um, species right now, but there's a research team that went down to Costa Rica and found a species of fungi. It's not a mushroom producing fungus, but it can consume um, plastic, 100% plastic, and it can do that in aerobic and anaerobic environments, which is insane. It's like it doesn't even need uh, air to continue to grow. And so what that means is it can do that in capped landfills, right? You could plant this fungus in a capped landfill where there's not really oxygen, and it would consume the plastic. Um, so there's some cool videos. There's a video of like a someone that converts plastic into an edible food, like an edible egg, um, on YouTube. That's pretty interesting. So definitely they can eat plastic, and it's a question of, how to do that on a scale, and how to grow these mushrooms, and that sort of thing.
There was a question that came in terms of the fruiting bodies. What does one do with the contaminated fruiting bodies?
Hmm. Yeah, I think it's depends on the contaminant. Um, the study that Paul Stamets did was showing that the fruiting body was clean, effectively. It was like there's nothing, after consuming oil as the primary food source, there's nothing toxic in the fruiting body. With that said, I'm not going to eat it. I wouldn't recommend someone else to eat it. So the mushrooms will just break down. They'll dry out, or they'll be consumed. They'll probably be eaten by a lot of beetles, and it's part of what draws insect life in. You have all these beetles and larvae that start feeding on the mushroom, and then all these larvae are growing out in the mushrooms, and birds are coming and pooping and dropping plant seeds, and the mushrooms become the um, like the mushroom itself, the fruiting body becomes this like attractant for life. Yeah, so that's what I was. And with heavy metals, you'd have to remove it, and there would be a toxic waste.
Can another question concerning working with larger agricultural sites, can bioremediation be used in large agricultural sites alongside crops? Have you ever seen that?
Alongside crops, like crops with herbicide or pesticide.
Say, and I may have Susan Zelenko, maybe if you can pull yourself off of mute and you can give a little more clarity to your question. I want to make sure that we have it documented correctly. I'm assuming it's like a cash crop. But Susan, you feel free to pull yourself off of mute and you can ask that question directly.
Hi, this is Susan. Yeah, the question, it's just after watching the documentary Kiss the Ground and seeing how soil can be completely depleted at times or uh, damaged, I saw what they were suggesting in terms of soil remediation on a large scale in agriculture. And I wondered if there were any attempts anywhere to use this bioremediation on a larger scale in the agricultural world, for example.
Mhm, mhm. I think, you know, two things come up for me is like one, I think one of the huge things with no-till agriculture is keeping the the fungal biome intact. You know, the fungal and the microbial biome intact, and keeping the mycorrhizal relationships with plants intact. So with agriculture, I think that's in fungi, that's a huge part. And also it's you know, very similar. We're adding in mycorrhizal inoculants or powders, or even just adding fungi, this sort of like spent substrate, can really bring soils back to life. You know, it's a huge increase in organic matter, and it's like worms and insects are really drawn to it. So I don't know of large scale projects where those are particularly being used, but I think the possibility for, you know, remediation of soils is huge, and it's a fungal health is a really important aspect to healthy soils and to growing healthy plants on soils, yeah, to just growing healthy plants. Because it's those mycorrhizal partners that help plants, you know, access nutrients, access water, break nutrients down. So when those are there, they're much, they're able to access a lot more, and in a relatively balanced and healthy way.
Now, there's a question about the mushroom crop itself after remediation, and this is from Dawn. If you successfully remediate on site, what do you do with the resulting mushroom crop? Will they die and become bacterial food? So what happens to that crop after remediation?
Yeah, exactly. So the mushroom. So is that like the mycelium? Yeah. So you have two things: the mushroom and the mycelium. We talked about the mushroom, which kind of dies and become a food source. And yeah, similar with the mycelium, eventually, when it doesn't have a food source anymore, yeah, it'll die, and other things will eat it. You know, other fungi. And what's interesting with uh, decomposition, fungi are like tiered. You know, it's like one fungus breaks down as much as it can, and then another, it unlocks all these compounds that now another fungus can break down. and that one breaks down all it can, and unlocks all these compounds that now another fungus can break down. So you have, primary, secondary, tertiary decomposers, and a lot of the white rot fungi are primary decomposers. So they're really at the starting chain of breaking down material. So they're going to eat a bunch of stuff, and they're going to unlock all these compounds that other fungi and other bacteria can also consume. So once they've eaten all they can, then it's going to go to the secondary decomposers, and they're going to start, eating the compounds that they can. So yeah, it's the mycelium eventually, I don't know, I guess dies, and other mycelium comes to fruition.
And in the remaining time that we have together, Willie, I don't know if you want to shift into this, but did you want to give a little bit of background or talk a little bit about the project itself that will be taking place in Springfield with our partners at Home City Housing and Gardening Community? Some of the things you'd like to do with the youth leaders there and the staff at those two sites.
Yeah, I mean, for me, I love mushrooms. I totally love mushrooms, and I want everyone else to love mushrooms, and just know how to work with them and the potential to work with them. So I think understanding mycelium is really, really vital, and understanding mycelium's role in creating healthy soil is really important. And the ability with working with fungi to be adding material. So much of our remediation and agricultural practices are like extractive, and the potential with mushrooms to be adding material to a site, building organic matter, breaking down pollutants, and how we can do that. So that's kind of what I'm really excited about, working with some of the youth leaders on, and exploring at the site of, how can we be creating more mycelium, and creating a healthy soil as we feed those mycelial networks.
All right, very good. Well, in our remaining time, if there are questions out there that we have not gotten into the chat, if anyone would like to take themselves off of mute and ask Willie those questions directly, we welcome that at this time. This is a very interesting topic, and I want to give a shout out to Carol Roselle, NOFA's education director. She's putting in some fabulous links from Natural Farmer. And another resource from David C. Johnson's research at Chico State University on fungal compost as a farm input. Those are in the chat. If you want a copy of those notes, you can just open the chat feature, click on the button with the 3 dots, and you're able to save this whole chat with all the wonderful links that have been put in there. But if there are any additional questions as we begin to wrap up, please feel free to take yourselves off of mute one at a time and ask Willie any of those questions.
Yeah, and I don't know if Andrew wants to add anything.
Yes.
Into the like what the project's doing, and where that's heading and stuff. That's welcome too, if you want.
Mm-hmm.
Yeah, I think we're all just blown away by the power of mycelium, and it's crazy. It's right underneath our feet. We don't always see it, and we think that it's. I've always thought it had to have the fruiting body of the mushroom. So that's amazing stuff, and just knowing that we don't have to get rid of our soil, and we can fix it, because once we get rid of it, then there's trash somewhere else. So we still feel responsible for it, and I think that's a big important part of this, is to take ownership of our spaces, and take care of them the right way. And I think that's a big part of this project that should be recognized as well.
I have a quick question about clover, how they fix nitrogen. Is there a special trick? I don't see any nodules in my clover.
I don't know. Is my best answer. I don't know how clover fixes nitrogen, and you might not be able to see the nodules, but my understanding of nitrogen fixation with plants is it's, like that symbiotic relationship with a fungal partner.
Mm-hmm.
But
I'm pretty sure it's a bacterial partner, Willie.
great.
And you can get clover inoculum from Fedco, Organic Growers Supply of Fedco. They have clover inoculum, and they also have Ryan Vetch inoculum. So it's sort of by which subspecies of legumes.
Thank you.
The inoculum is the bacteria.
Right.
Yeah, it's I think it's Rhizobacterium or Rhizobacillus or something. I have to look it up, but I'll look it up and drop it in the chat.
Thank you.
I like Jim's comment. Fried red clover flowers taste like fried dough. Nice.
For those who may be having trouble actually getting the chat notes, I just put my email in the chat. You can send an email to me. I've saved it, and I'll send you a copy of the file for your record so you can refer to those, to these wonderful links. Willie, any closing remarks or comments? And same thing to you, Andrew, as we begin to wrap up.
There's this other question here. Can NOFA set up a myco hotline for future questions? I love that.
I like that one.
I'm gonna call my customer service line the myco hotline now. Oh, so good. Thanks for that. It's great.
Well, Willie, you could mention your website and your information if people do have additional questions for you.
Yeah. If you have more questions or want to learn more about mushrooms or work with mushrooms, fungially.com is my website, and you can email me or call me. All my info is on there. And yeah, would love to talk more, and I love hearing about microremediation projects that are happening, and connecting people with mushroom farms. I'm pretty well connected into the national mushroom farm scene, and a big part of starting microremediation projects is just sourcing mycelium. So, mushroom farms are a great source for that, and just to get spent mycelium and be inoculating materials. So happy to help with that sort of sourcing.
Yeah, Willie, have people called you or ordered kits for remediation, or do you notice it's just mostly for like food?
Yeah, I've worked with people buying spawn for remediation projects, so oysters and wine caps both. There's great like home cultivation methods where you can be expanding out mycelium on wood chips or straw, to be implementing. And particularly wine cap is really easy to grow, and you can get hundreds of pounds of mycelium pretty easily. Yeah.
There is one more question that I think will be very key for those backyard growers like myself. This is a question concerning homeowners. What can they do to remediate their land and possibly remediate using this technique?
Yeah, I think it's the first thing is always what are you remediating? What's the thing that you're trying to break down or add? And I think just like bringing more fungal diversity, whether that's growing mushrooms or contacting your local farm and getting spent mushroom substrate from them, and adding that into soil. Growing wine caps makes incredible soil. Just getting wood chips, if you just get wood chips on your property, mycelium's in there. It's like you don't even, if you're just looking to build organic matter or remediate stuff, you can probably just have organic matter that's growing mycelium and put that in the soil. Have that on your yard. Add material. And the fungi are, yeah, totally, the fungi are all around us, you know, and we don't know. There's so many species that aren't studied, you know, so it's not like, oh, we have to have this one species. You're probably going to get a lot of the benefits from native fungal species, and you won't be able to necessarily eat the mushrooms, but definitely be building organic matter. And if you want to, you know, if you have specific petroleum products that you're trying to break down, oyster mushrooms are awesome. And isolating that contaminated material in a bin or something and growing oysters there is fantastic.
Mhm. All right. Excellent, excellent. Well, thank you so much, Willie. As we wind down and begin to close out, this has been such a great class, and we are so grateful to you first for the webinar and then for the continued work that you'll be doing with us in Springfield. And if you all would like to follow the story of our bioremediation project, please check out, I'm gonna put them on the spot, I have to, but please check out Andrew's article that'll be coming up in subsequent newsletters about the story of our bioremediation projects at Gardening the Community and Home City Housing. Definitely, this is a great study, and we're very happy to have Willie as one of our consultants. And I wanted to add, I missed one of our consultants. I'm really appreciative to Carol putting this in the chat for me as a reminder. We are also joined on our consultant team by Naveen Ishmael. She is with Smith College's engineering department. Her and her wonderful graduate students have already installed mycelium blocks, I believe, Andrew, at Home City Housing's plot. And so we're just looking forward to uncovering those soon. And we're hoping to have her join us on subsequent webinars and online events. So again, thank you to everyone for joining us. Do not forget, December 1st is part 2. This is where you get to hear from the on-the-ground work from Andrew, from Ibrahim Ali at Gardening the Community, some of the youth leaders from Gardening the Community and Home City Housing, as they talk about the first year, our first year of work with this grant. It will be at 7 p.m. You can reach out to Doug Cook, who is the educational events coordinator. He'll be hosting that event on December 1st. So please, you don't want to miss that. This is where you get to see what Willie is talking about live and in living color in action on the ground. So I do invite everyone to come back and see that. Also, this is a big one. Our winter conference will be virtual. It is January 8th through the 10th, 2021. You don't want to miss it. Please go to our website, nofamass.org, to register. We have some wonderful workshops all about feeding the Commonwealth, food security for the Commonwealth. And we'll be joined, one of our keynote speakers is Greg Watson, former State Secretary of Agriculture here in Massachusetts. That's something else you don't want to miss. And again, if you want more information about Fungi Ally and what Willie is doing, his amazing work, please go to his website. So with no more from myself, on behalf of Andrew, my partner coordinator, and Carol Roselle, we thank you for joining us. You all have a good night, be safe, be well, and we'll see you 2 weeks, December 1st. Thank you all so much.